speaker is Gerome Breen, Professor of Psychiatric Genetics at KCL in London. His research interest is understanding biological basis of psychiatric diseases using a genetics-driven approach, and he's going to bring an academic perspective on the challenges and opportunities in the field. So over to you, Gerome.
Thanks, Alessandra. So I'm going to give you a talk today. In essence, this is some of the ideas that I'm thinking about at the moment in terms of what we're possibly going to be able to do in the next few years. So first of all, as Pat Sullivan outlined on day one, the PGC is currently applying for its 4th round of funding. It's highly likely that the progress that we've seen in what have been just a handful of disorders to date, but a growing number of them, will be replicated in every psychiatric disorder over the next 5 years. So the picture that we currently see in schizophrenia and depression will maintain in virtually every psychiatric disorder in 5 years' time. So we'll be left with not just hundreds as there are now. There's probably around 500 loci now. We'll probably have several thousand loci, both shared and distinct across psychiatric disorders within about 5 years. We know that in the best powered psychiatric GWASs to date, we can effectively capture the targets of known drugs. So this is work from a postdoc who worked in my group, Helena Gaspar. She now works in industry. And she just made this visualization in terms of if you rank known drug targets by their significance in GWAS, how many of them are captured as a percentage rank. And essentially, as you can see, you got a very nice illustration of the power of GWAS here. So from the PGC 1 schizophrenia with 9,000 cases to PGC 2, sort of 14,000 cases, and PGC 1 plus Sweden, 14,000 cases, and PGC 2, 36,000 cases. Gradually, the GWAS got better and better at capturing known drug targets, known drug mechanisms and pathways. So, in other words, the other information that is in the GWAS then is being suggested as, potentially therapeutic if its mechanisms can be addressed. Going on from that, we see many of the single genes that you can identify from GWAS. So, it's not always possible to identify the gene that underlies a GWAS hit, but actually you can do that for a significant minority of the hits. And we do see good evidence for some known drug targets. Now, the only thing is that many of the drugs that we can currently, drug targets we can currently tag are known to bind drugs that are used to treat cancer, for example, and other things. So, but I think it does show that, when we get well-powered GWAS, we can find things. But I wanted today, instead of focusing on, genes as targets and so on, I wanted to talk more about what else can we learn from psychiatric genetic approaches. And so one thing that's been emerging, and Andrew Grotzinger is going to talk about this a little bit later, is that at the genetic level, psychiatric disorders are complex. They do share a lot of their genetics as they share a lot of their symptoms, but just as with their symptoms, their genetics do cluster. So psychiatric disorders cluster into subgroups. So in this simple cartoon here, driven by genetic correlations, you can see that disorders with compulsive behaviors cluster together, as do early onset neurodevelopmental disorders, as do a sort of loose clustering of mood and psychotic disorders. However, I think you might say here that maybe major depressive disorder lies somewhere in between all three, but I think there is some evidence of distinction. We've also been sort of delving more into the phenotype. So this is work by Jonathan Coleman, who you may have seen tweeting about the conference. He was a postdoc in my group, now is a lecturer in our center, and he worked in this study where we looked at the different subtypes of mood disorder. So all the way from subthreshold depression, so people who have one of the two core symptoms of depression but don't quite meet the full number of symptoms required for a diagnosis, up to single episode depression, recurrent depression, bipolar disorder type 2, type 1, and schizoaffective disorder. And this diagram reflects the genetic correlations between these disorders. And what was really, really interesting to see was that although recurrent depression had the best heritability of any of the depression subtypes, essentially subthreshold depression, single episode depression, and recurrent depression all had really high genetic correlations with each other. So they had, as it were, they pretty much had the same genetics, even though their heritabilities vary. And what we think this indicates then is that the genetic load essentially in subthreshold depression is less than the genetic load in recurrent major depression, but it's essentially the same genetic variants, the same genes are represented. Equally, we've been looking at genes and environment. So some of the most disputed work in psychiatric genetics has been on single gene candidate gene environment work. And it's long been a desire of people who work in the field to acknowledge the role that the environment plays in the etiology of these disorders, and to examine how that affects genetics. So what we did in this study was, using UK Biobank, was to conduct a systematic analysis in around 26,000 depression cases, looking at depression polygenic risk score by trauma interaction. And we were able to show a couple of things. Yes, the polygenic risk score robustly interacted with trauma in a multiplicative way. But this paper also showed that depression exposed to trauma seemed to be more heritable than depression not exposed to trauma. And this is essentially a very large study with 26,000 cases. It's not yet at the scale at which we can discover single variants, but we hope in the next couple of years that we will be able to conduct sufficiently well-powered studies of 100,000 or more cases to discover variants that underlie gene-environment interaction. So one more thing then, just generally to cover, what can we expect in the next few years? So the recovery of, so this paper here is a paper by Peter Vischer's group. It's up in bioRxiv. I think it will be published soon. In this paper, they use data from the TopMed sequencing consortium. So I think they had 20-some thousand people with sequencing data and height information. And they estimated heritability using their GCTA Greml package. And they were able to estimate heritability using whole genome sequence data of around 80% for height and around 40% for BMI. So in other words, almost totally recapitulating the estimates from twin studies. And they comment that they were able to do this because they were actually better capturing lower frequency variants that may actually contribute a lot of the genetic variants. They do, however, make the point that whole genome sequencing sample sizes, in order to reliably detect single variants underlying this type of signal, will need to be as large as whole genome sequence studies. As large as genome-wide association studies. So in other words, people will need to whole genome sequence hundreds of thousands of cases. But there is, sorry, let me try to advance. Okay, so, but there is another thing that may happen in the next year, which may get us part of the way there. So this plot here is the from the Schema Consortium. So it's an exome sequencing consortium for schizophrenia. And here you can see the odds ratios for single genes or genetic variants, for rare variation, rare variants that have been identified, and for the variants that have been identified from GWAS. Now you can see, I think here, that there's a gap from about 2% or so down to about 0.1% in allele frequency, and the whole exome sequencing or whole genome sequencing studies that exist aren't big enough to detect those variants at the moment in terms of their effect size, and the GWAS imputation hasn't been good enough. However, in the next year, imputation is going to be revolutionized because of the TopMed imputation consortium. So there's an interim release now of, I think, about 65,000 whole genome sequences from diverse ancestries. And this will give a dramatic improvement to our ability to impute extremely rare variants. So actually using existing GWAS data or the GWAS data that's going to be coming in the next year or two, we should be able to get to detect many more rare variants. And hopefully then that will also invigorate or target identification. I just wanted to also then essentially plug a paper from Topher Hubel, who's the co-organizer of this conference. So Chris actually declined the opportunity to speak because he wanted to organize everything behind the scenes and make sure everything was running smoothly. But this is work that he did and is published in Nature Communications, looking at the genetic correlations of psychiatric traits with body composition and glycemic traits. And I'm not going to go into the ins and outs of this paper, but essentially here we were looking at the relationship between body composition and psychiatric disorders broadly defined. Topher identified that there were two groups of disorders. One group of disorder that was associated essentially with a smaller body size, decreased body fat, etc. And this included anorexia nervosa, OCD, and schizophrenia. And another group of psychiatric disorders, ADHD, alcohol dependence, insomnia, which seemed to be genetically correlated with larger body size. Moreover, Chris was able to do some very interesting Mendelian randomization analyses, looking at psychiatric disorders as an exposure on an outcome, body fat. What was quite interesting here was that, okay, the biggest effect on body composition is education. Okay, so GWAS of education years has a fairly large effect in terms of percentage body fat reduced. Anorexia also has a relatively large effect, but if you look at other traits, the other psychiatric disorders that show a signal are OCD and schizophrenia. Now, OCD makes sense because of compulsive behaviors and actually it's close phenotypic relationship with anorexia. But schizophrenia is interesting. So the typical picture of schizophrenia is of obesity and metabolic syndrome. But it does appear genetically, people with schizophrenia may be predisposed to smaller body size, lower body fat. And this leads then to perhaps another type of conclusion that we can draw. So do the current drugs for schizophrenia actually target its core biology, not only, let's say, in the dopaminergic system, but also in the side effects they cause of weight gain and metabolic syndrome? Okay, so switching gears, I just wanted to cover a couple of things. So polygenic risk scores have become quite famous now and are being used in a lot of academic studies, but relatively few clinical studies. People are most familiar with the schizophrenia polygenic risk score, it accounts for about 7% of the variance in on the liability scale, on the population scale. In depression, the polygenic score is not as strong, but is getting more useful. So currently it accounts for about 2% or a little more of the variance in case-controlled status. People have verified that polygenic scores are higher in recurrent cases, more severe cases, and early onset cases. Other fields like coronary artery disease and breast cancer are advancing in terms of their use of polygenic risk scores. Not without controversy, of course, but it does appear that these fields may be the first to make use of polygenic risk scores in the clinic. Notably, polygenic risk scores were invented in psychiatry. Thus, what is holding back their clinical use? Well, we think it might be the lack of essentially pre-genotype cohorts in which to carry out intervention studies in clinical trials. And so I want to tell you today about the genetic links to anxiety and depression study, which I run with my collaborator, Talia Ely. GLAD is designed to be a recallable, recontactable cohort of 40,000 participants with either clinical depression or anxiety. Participants are consented to recontact by phenotype or genotype, or in other words, clinical variables and or polygenic risk scores. GLAD is an entirely online resource. We do recruitment online, and we do questionnaire-based assessment. We launched GLAD in September 2018, and as of today, we have about 23,000 people fully enrolled in the study. Actually, the lab that receives our samples is currently closed. It's become one of the major COVID testing centers in the UK. So we actually think that we might be over 24,000 cases at the moment, but we won't know until the whole COVID situation calms down. GLAD is a national cohort in the UK. So we collaborate with excellent people in Northern Ireland, Prof. Sherry Armour, Scotland, Prof. Andrew McIntosh, and Wales, Prof. Ian Jones, to do recruitment in all 4 nations of the UK. This density map shows where we are recruiting. So unlike studies such as even UK Biobank, we actually have broad national recruitment of cases. And in fact, we have an enrichment of recruitment in areas that are of lower socioeconomic status, which is, I think, highly relevant. These are the current numbers in GLAD. Now, one take home from this is that we have a wide range of diagnoses represented, but also we have a very severe cohort. So 82% of the GLAD cases have recurrent major depressive disorder. And as such, then this online-based recruitment that we've been doing seems to be yielding quite severe cases. GLAD is designed to enable research into all different types of psychiatry and genetics. It is possible to recontact and select individuals on the basis of their polygenic risk score, their clinical symptoms, or their demographics. And if any investigators want to conduct a study using or recruiting from GLAD, please email us. We promise you a swift review of your proposal. The cohort is set up to be directly recontactable. I just wanted to finish by thanking my group. So I also run a genomics and biomarkers core facility. We are paused at the moment in our work, but it is the UK's largest Illumina genotyping facility. Several of my team members, pictured here, are currently volunteering in the NHS. Also want to acknowledge my research group. Here we are on our research retreat last year in Ireland, actually in Southwest Ireland, where I'm from, on top of Ireland's highest mountain. And finally, I'd like to thank the audience and acknowledge the GLAD team, the bioresource team, who've been doing all our recruitment efforts. Okay, thank you very much.
Thank you, Gerome, for the excellent talk. So we have time for one question, which is from Alex Atum, which is, how do you think that misdiagnosis would influence genetic correlations? And is there any evidence of these in the PGC?
Okay, yes. So I think we have to realize that there's considerable type 1 error in the diagnoses especially common psychiatric disorders, as in they can get misspecified. And we also have to recognize that there's ascertainment in our cases and controls that could also influence our genetic correlations. So I think then it's that would, I think, influence things. There's one example I would cite. So 23andMe depression has a slight positive genetic correlation with IQ, but clinically ascertained depression has a significant negative genetic correlation with IQ. And that gives you an example of how ascertainment might bias your polygenic risk scores.